Good day, good afternoon, good evening to all of you watching. And in this lesson, we are going to talk about inorganic compounds that are essential for the functioning of human. So, a quick recap for the last section, we discussed about chemical reactions and enzymes, how those bonds are formed, how those energy is managed inside the body. Now, in section 2.4, we will now focus on inorganic compounds that make those reactions possible. So, inorganic compounds may seem simple, but they are absolutely very essential for human life. So, in this section, a quick overview so that if your topic is here, you can continue watching. So, in this section, we will focus on dehydration, synthesis, and hydrolysis. We will also focus on the dissociation of sodium chloride and water, as well as acids and bases, and the pH scale. And together, these concepts explain how molecules interact in water, um, which is water is the primary environment of the human body. As you know, water is like 70% of our body. So first, let's talk about dehydration, synthesis, and hydrolysis. So let's begin with this. So these are two opposing chemical processes that are fundamental to biology. Living organisms are built from large molecules, which are also known as macromolecules. You probably tackled this in 12th grade, which are like those carbohydrates, proteins, lipids, and nucleic acids. And these large molecules are formed from smaller building blocks of what we call monomers. So carbohydrates, monomers are I forgot. Proteins has a monomer of amino acid. Lipids has a monomer of fatty acids. Nucleic acid has monomers of DNA. Yeah. So we're gonna talk that on the next lesson. Uh, the macromolecules. So first, let's talk about dehydration synthesis. So in dehydration synthesis, um, two monomers are joined together to form a larger molecule. So the word dehydration literally means the removal of water. You know, when we are dehydrated, we are in need of water. So, and if we are dehydrated, we have less water in our body. So during the dehydration process, one monomer loses a hydrogen atom, and the other monomer loses a hydroxyl group. And these combine, this hydroxyl group and this hydrogen atom will form into water, which is H2O, as we know. And a covalent bond is formed between these two monomers. So that's the dehydration process. And these monomers are linked by covalent bonds. And this reaction is used to build disaccharides and polysaccharides from monosaccharides. Oh yes, the monomer of carbohydrates is monosaccharides, and it also this process builds proteins from amino acids. It also builds lipids from fatty acids and glycerol, and it builds nucleic acids from nucleotides. It's not DNA, it's nucleotides rather. So yeah, and dehydration synthesis is an anabolic process, meaning it builds larger structures and requires energy. So as you can see, um, these two monomers forms into a forms into a water molecule, and these monomers are linked by covalent bond with this oxygen atom left. Okay, so next is the hydrolysis. So this is the opposite process of dehydration synthesis. The meaning of hydrolysis is splitting with water. So in hydrolysis, a molecule of water is added. Then the water splits into hydrogen, into an hydrogen atom and a hydroxyl group. And these components break the covalent bonds here between the monomers. So as you can see, this is just the the reverse process of dehydration synthesis. Hydrolysis reactions are essential for the digestion of food, for breaking down stored energy, for recycling circular components. So, as you notice, observe that dehydration synthesis, its functions is to form different bonds. I mean, it's to form different molecules to form a much larger molecule because that's the that's the process. It forms molecules, and the uh, the the hydrolysis in real life situations are used for breaking down substances because the process breaks down. As you can see, this covalent bond is broken down to form these. And as I said, digestion of food, it makes breaking down of molecules, the breaking down stored energy, breaking down. So you notice. So dehydration, it builds up. Hydrolysis, it builds down. Um. So for example, in hydrolysis, starches are hydrolyzed into glucose. Proteins are hydrolyzed into amino acids. So as you can see, it's just like the reverse process. As like amino acids are building blocks of proteins, but when hydrolysis occurs, proteins are hydrolyzed into amino acids, just like that. So together, dehydration synthesis and hydrolysis allows the body to build and break down molecules as needed, which is needed for balance, for maintaining balance and homeostasis. Next, let's talk about dissociation of sodium chloride in water. So let's look what happens when ionic compounds like NaCl dissolve in water. So we will use sodium chloride as an example or table salt. So table salt is held together by ionic bonds between Na plus ions and Cl minus ions. So however, when sodium chloride is placed in water, so this is sodium chloride is placed in water. So these are what this is a water molecule. This is a sodium ion. This is the crystal of NaCl. It contains sodium and chloride ions. So when chloride ions, as you can see, it surrounds these. Okay. So I'll do the proper discussion first before I um I explain that. So so when sodium chloride is placed in water, it does not remain intact because we know that water is a polar molecule, so it can interact with charged particles. But when sodium chloride dissolves, the slightly negative oxygen end of water is attractive to end Na. So hydrogen here, I mean oxygen here tends to have a negative partial charge, slightly partial charge, right? And Na plus ions is positive since it's Na plus. So the oxygen atoms, since unlike charges unlike charges attract, these forms a little attraction here, okay? Okay, so as you can see, they repel, but as you can see, it's different here. But when the end of the water is focused on hydrogen now, because it forms because hydrogen tends to have a positive partial charge, and it attracts with chloride ions, since chloride is negatively in charge. So these attractions pull the ions apart, which is what we call as dissociation. So each ion becomes surrounded by water molecules, forming what is known as the hydration sphere, which this hydration sphere keeps keeps the ions separated and suspended in the solution. So this is very very important, as free ions move move and conduct electricity, and electrical activity is essential. Our body produces electricity for nerve impulses and muscle contractions. Um, it's also important for electrolyte balance, depends on dissolved ions. So sodium chloride in the body exists not as these crystals but as these free sodium and free chlor- chlorine ions that are surrounded with water. Okay? So next, let's talk about acids and bases, which is a major role in chemical balance. So, so acid is so take note that acids are substances that releases hydrogen ions when dissolved in water. Like for example, HCl, we have what we call hydrochloric acid. It dissociates into H plus and Cl minus ions. Hydrochloric acid is found in an example is that is found in the stomach, which is essential for digestion. And acids increase the concentration of hydrogen ions in the solution because it releases oxygen ions. It releases hydrogen ions when dissolved in water. So I don't know if I said oxygen on the first part, but as I said, acids increase the concentration of hydrogen ions in the in in, in the solution. Hydrogen ions. Always think acid is associated with hydrogen ions. Okay, next let's talk about bases. So bases are substances that releases hydroxide ions. Acid releases these H plus ions, hydrogen ions, while bases releases hydroxide ions or OH minus ions, and or it accepts hydrogen ions. So an example of that is example of that is sodium hydroxide or NaOH, and it dissociates Na plus and OH minus. So since it has OH minus, it's an example of a base. Since a base releases hydroxide ions or OH ions, and bases reduces hydrogen ion concentration. So remember, again, I keep saying this, remember that acid releases hydrogen ions and bases releases hydroxide ions, as well as um, acid increases the concentration of hydrogen ions. Meanwhile, um, meanwhile, the hydroxide ions reduces the hydrogen ion concentration. So, importance of the body. Hydrogen ions are very, very reactive. Too many hydrogen ions can break chemical bonds. It can alter protein shape, which can be very dangerous. It can disrupt enzyme activity. Meanwhile, too few hydrogen ions can also disrupt normal cellular function. This means that the body must regulate acid-base balance to maintain homeostasis. Too much or too low is very very kind of dangerous. So we need to regulate a balance of that throughout our body. Next, let's talk about the pH scale, the last part. So to measure acidity and alkalinity, we use the pH scale. So the pH scale measures the concentration of hydrogen ions in a solution. So it ranges from 0 to 14. Um, pH pH 7 is neutral. Below 7 is acidic. Below 7 is acidic. Above 7 is basic or alkaline. So each step on the pH scale represents a tenfold change in hydrogen ion concentration. This means that small changes in pH represent large chemical changes. Okay, here's some things to take note about. Ala, um, pure water has a pH level of 7. So that's the neutral. And blood, the pH level of blood is between 7.35 and 7.45. So as you can see here in the picture, it's the average of that. Okay, and acidosis occurs when the pH let's talk about blood problems and an imbalance of blood, and when blood pH is below 7.35, that's what we call acidosis, from the word acid. Acid means the pH level is below 7. And alkalosis occurs when the pH of the blood rises above 7.5 from the word alka, meaning alkaline, which is above 7 is alkaline. That's what we call alkaline. Even slight deviations outside the 7.35 and 7.45 range of blood can impair enzyme function. It can disrupt nerve signaling, and it can even cause illness or death. So, maintaining the blood pH level is very, very crucial. So, of course, our body doesn't want that. So, therefore, our body uses buffers to resist the changes in pH. So, these buffers can absorb excess hydrogen ions and releases hydrogen ions when levels drop. So it's like it kind of balances the pH level of your body. And the most important buffer system in the blood is what we call the carbonic acid bicarbonate system. So take note of that. So buffers allow the body to maintain a stable environment despite constant chemical reactions. So our body is very, very amazing, isn't it? So that's it for this lesson. I'll give you a heads up. This is actually the last lesson for this chapter, organic compounds essential to human functioning, where we will talk about monosaccharides, disaccharides, polysaccharides, triglycerides, saturated and unsaturated fatty acids, lipids, amino acids, the peptide bond, proteins, enzymatic reactions, nucleotides, disaccharides, and ATP. It's a very, very long topic. We might actually even take an hour to do that. But anyways, that's it for this lesson on inorganic compounds essential to human functioning. So I hope you understood something in this lesson. I'll give this video a thumbs up if you understand something. Give this video a thumbs down if I explained bad. So that's it for this lesson. Thank you. Have a nice day and see ya.